Hi everyone. I'm Youngmo Kim, and I'm a scientist at PNNL, and I'm working on metabolomics research for a long time here. And before starting my talk, actually the EMSL summer school, we did a small survey with all the participants, and then I realized that some of the participants are not familiar with the chromatography or mass spectrometry at all. So I thought that I want to be a more fundamentals for my talk because I know some of you already have experience on the chromatographic separation or mass spectrometry measurement, but some are not. So today my talk is more fundamental. So if you are not familiar with this mass spectrometry or chromatography, but I think you will be aware of how this works for metabolomics research. So today my talk is what is chromatography and what types of chromatographies are used for metabolomics research, and then what is a mass spectrometry, and also what types of mass spectrometry are used for metabolomics. And Natalie briefly mentioned about the difference between the targeted metabolomics and the global metabolomics discovery. And then I will cover a few subtopics for mass spectrometry-based metabolomics research. As Natalie mentioned, because some of the content might be overlapped a little bit, but I think it's good to remind you guys to better understand metabolomics and metabolome. So metabolite is a metabolic intermediate or end product from certain metabolism. And the total pool of metabolite present within an organism or cell or tissue is called the metabolome. That is just overall pool. And they have a huge variation in their molecular weight, hydrophilicity, hydrophobicity, or acidic or basic, volatility, chemical stability, and many other factors. And then the important thing, what Natalie mentioned, they never exist same amount in the system. So if they exist same amount, it might be easier to measure all of them, but the reality is not. So for example, if we analyze human plasma samples, glucose is the most dominant peak compared with others. And if we analyze urine samples, urea is the biggest peak compared with others. So it's really hard to get everything captured. So how can we measure them accurately then? The answer is the combination of a chromatographic separation and a mass spectrometry measurement. That's why I'm talking about this tool for metabolomics research. So traditionally, the chromatography, you may know this simple experiment. So if we put plant extract or some other mixtures in a paper, and then if we put that paper into solvent or water, when the liquid moves to the top, we will see some separation of the components. So like this, the chromatography is a tool for the separation of a complex mixture. And traditional metabolite analysis has been done with several steps of purification or cleanup. Then we can just focus the analyze what we want to measure. However, metabolomics approach, we need a minimal cleanup step, just removing salts, for example, because if we do a lot of purification or cleanups on samples, we will lose some metabolites or some lipids. So that is a kind of a trade thing. So we need to minimize our cleanup, and then we will get a lot of metabolites. However, in that case, some interference, the results might be not 100% accurate because there are some interference in between all the metabolites. And gas chromatography and liquid chromatography are the most common equipment for separating metabolites before mass measurement. So that is what I will talk about in the next slides. And sometimes metabolomics or lipidomics samples can directly inject to the mass spectrometers without this separation step. We call it direct infusion or shotgun analysis. So if we have a very high resolution mass spectrometers, probably this can help without spending a lot of time for separation, because usually liquid chromatography or gas chromatography takes a quite significant amount of time to separate. So there's some people still use this direct infusion or shotgun analysis. What is chromatography in reality is a separation of metabolites by the interaction between the mobile phase and the stationary phase. So mobile phase is something running through this column, and stationary phase is located inside of this column. This could be coated inner surface or packed something. And if we see this simple animation, we can separate the complicated mixture by their chemical property. So some metabolites are retained by the stationary phase. And then that is the chemical properties, the molecular weight or volatility, acidic or basic or hydrophilic or hydrophobicity. So there are a lot of interactions between stationary phase and the metabolites. So if the mobile phase is liquid, we call that is a liquid chromatography. And if the running mobile phase is gas, then we call that is a gas chromatography. So this is a simple scheme of gas chromatography, and there is a gases here. It goes to the old system until all the analytes reach to the mass spectrometer. And the fundamental thing in gas chromatography is using heat. So when we inject sample, and then if we increase the oven temperature from 40 or 50 Celsius degree to about 300 Celsius degree, and small molecules will vaporize easier and faster, and then will pass this long column and then will go to detector. And then bigger molecules, they will be evaporated later, and then we will see that signals in later time point. That's the very fundamental scheme of gas chromatography. So a lot of gases can be used for the mobile phase, helium gas or nitrogen or hydrogen, but mostly we prefer to use a helium gas for the gas chromatographic separation. And also there are many different types of GC columns, because I will explain a little bit more detail about this, but based on the purpose, we can choose the column what we want to use. So non-polar column to polar column, we can choose. But in general, global metabolomics, we want to use a non-polar column, and volatile metabolites, we prefer to use a polar column. And what is the difference between the polar column and non-polar column? So this is the product table from Asian Technology, and they separated 3 different groups: the low polarity or mid or high polarity. So the right one, this is just cut of the GC column, gas chromatography column, and the carrier gas, for example, helium gas, run through this column. And inside of this, there are some material are coated in a surface. So if we put this polyethylene glycol or similar structural polymers inside of this column, there are a lot of dipoles and electronegativity of oxygen. So it is relatively became polar. And if we coat the inner surface with this methyl siloxane with a phenyl group, this column will be a non-polar. So non-polar means it interacts with the non-polar compounds, and polar means they interact with the polar metabolites. And let's move to the liquid chromatography. It's the same thing, but as I mentioned in the previous slide, the mobile phase is not a gas. This is this time this is a liquid. So in general, there are two different kind of a concept of page separation. So a lot of people may heard the reverse page or the normal page or C18 or Healy. And let's talk about the reverse page first. So yesterday, the lot of proteomics side, the Christine, Yuchen, or Ibet mentioned about separation of peptide, the tryptic peptides using the reverse page column C18. That is worked using this aqueous mobile phase, and then we will give a gradient. So at the beginning, the all mobile phase is a polar eluent, and then gradually we put more organic mobile phase. So if that's the case, how we separate metabolites is so if you see here in the reverse phase, polar metabolites elute early and non-polar metabolites elute later, because the non-polar metabolites, they will interact the stationary phase, because this stationary phase is non-polar. So they want to stick each other, and then we put more organic solvents, and then they will be separate, and then it will go to the end of the separation. And so this is called the reverse phase. And then if we see normal phase, and this works opposite way. So if we put normal non-polar solvent eluent first, and then eventually we put gradient to polar eluent like aqueous, methanol, or water like that. If that's the case, we will see the separation in reverse order. So non-polar metabolites elute early and polar metabolites elute later, because the stationary phase is polar material, so they still have an interaction each other. So again, this reverse page is C8 or C18. The packing material has a very lipid, the C8 or C18 is a hydrocarbon chain, so it's like a oil or lipid. So they want to interact with the non-polar metabolites. And helic is amide column, and they interact with the polar metabolites, while the non-polar metabolite just pass the column. So that's how separations are happening through the liquid chromatography. And I want to show us a couple of examples of chromatographic separation. So this is the result from a separation of a fatty acid and methyl esters. We use this mixture every day for GC analysis. So as you see, the C8 is the carbon 8 octanoic acid and methyl ester. So this will go to the separation, and we have a mixture like this, and we can separate them by the molecular weight and their dimension. So if we run this mixture every day, we can check our column separation works fine for analysis of real samples. So this is a confirmatory analysis every day, and this is a real sample. This is intracellular metabolites from Pseudomonas putida, and we extracted the metabolites, and we can see like a lot of peaks. So each peak represents a peak of a metabolite, but as Natalie showed in the previous slide, some of them are still mixed, the combination of 2 or 3 separate different metabolites. That's we call co-elution. And so further data processing will separate them by the processing step. So that's how we can measure a lot of different metabolites from one dimension of separation. And this is an example of the liquid chromatography of lipids. And this is a Rhodosporidium toluroides is one of our ABF host strain. It's an yeast-like organism, and we use this for producing a lot of hydrocarbon-related or some unique hormone or pheromone kind of compounds. So this is so far I covered the chromatography for liquid and gas. And let's move to the mass spectrometer. So mass spectrometer is a kind of a very exciting or interesting instrument to analyze some chemicals. And a very long time ago, the J.J. Thomson is a British guy, and then he invented and then he did this cathode ray experiment. And then he found that charged particle will be deflected under the electric field. So this is a cathode ray, and then without any electric charge, it goes straight. However, if the environment is under the electric field, and then the direction is deflected, and then later he came to know that it is proportionally to the molecular weight of the compound. So if bigger molecule, heavier molecule, or lighter molecule, the deflection rate is different. So this is a very fundamental of the mass spectrometer. After this invention or finding, there are a lot of great scientists improved this measurement of the particle. So, and then we need to know what is M over G values. You probably saw a lot of M over G values yesterday, and that is about the mass to charge ratio. So M is a molecular weight of the compound, and Z is their charge status. So there are simply three possibilities. So if the molecules are in electron ion impact ionization, the molecule will lose one electron, and then it converts to the one charge positive ion. And then electron spray ionization, there are two modes. One is a positive, the other is a negative. So if the molecule can get a proton, then overall the molecular charge state is +1. And if the target metabolite is an acid, then they easily lose one proton and dissociate to for example, lactic acid converts to lactate. So that process can make that molecule is a negatively charged one. So if we just make these gases, so methane, nitrogen, oxygen, carbon dioxide, ethanol, so if we make this mixture and if we inject the mass spectrometer without separation, we will see these peaks. So methane peak will be shown at 16, and the nitrogen gas shown up in 28, oxygen is 32, and carbon dioxide 44, and ethanol is 46. So we can accurately measure the molecular weight of the target compound. So the dimension in the x-axis M over G values, and then we detect their molecular weight accurately. And what is a mass spectrum? So, the light spectrum can be seen through a prism. So light comes through this dimension and the deflection happening, and then we can see the different colors, different light based on their wavelengths, and we recognize the colors. It's the same thing. And the mass spectrum is at one point during the chromatographic separation, if we click this chromatogram, and then we can see the M over G values at that moment. So we can separate them by their molecular weight over charged ratio. So in general, GCMS, or LCMS, we mostly just see one charged status. But yesterday, like top-down proteomics, we see highly multiply charged protein because their other moieties can get additional protons. So we can see this mass spectrum at some point. And mass spectrometers, there are 5 components in general. So the inlet, the sample introduction from gas or liquid, and then there is a ion source for ionization, and we have to have a mass analyzer to separate them by the M/G values, and then we will detect how intense they are, and then the further data systems are working in computer. And these 3 parts should be in vacuum conditions. That's always I put this color. And there are many different types of ion source, ionization, and mass analyzers. So I cannot cover everything here, but I just will add a couple of things in the next couple of slides. Hard ionization is a little bit stronger ionization. So it usually employed in the gas chromatography mass spectrometers, GCMS. So this ionization process, the gas molecule will pass through the ion beam, the electron beam from top to bottom. And this electron beam has an energy. And then it will take additional electron from the molecule, and then the remaining molecule will be positively charged. So the butane, for example, if this molecule passes the electron beam, and then it will be ionized. So there are a lot of possibilities. So if the whole structure survived, it's the molecular weight is 58, and then you will see the 58 peak in the mass spectrum. So you see this 58. And if this electron ionization is hard, and then there are partial breaking down can happen, and then it loses 1 methyl group, and then the molecular weight is 43. And then this part is positively charged, and then we can see the 43 peak here. And then if this molecule is breaking at the middle, then either side 28 will be possible. So here you can see 28. However, you can also see 27 and 29, because during this fragmentation, there are additional proton can be located to the left side or right side. So there is plus minus 1 is possible. And then the last part, 1 methyl group is 15. So all the mass spectrum can be interpreted like this way. And this pattern and this relative ratio is very unique chemical property. So if we don't know what is our target, and then we inject the sample to the machine, and we got this spectrum, then we know that that gas is butane, because 58, 43, and these fragment patterns are very unique. And then our library says it's butane. This is how the hard ionization works. And for the metabolomics, for example, this is a citric acid, and then if we do the chemical derivatization, and then that also undergo the same breaking down under the electron beam, and then it will generate a lot of fragments like this. And then ideally we can identify which peak is what. So if we need, we can focus on one certain fragments, and then we can do more kind of isotope tracing. So if these fragments got isotope labeled or incorporation, and then we will be able to explain how much they got labeled in which fragments. So this is the GC hard ionization. And then that was so strong. However, from this slide, we call this soft ionization. So we don't do harsh condition like electron beam. And this is a tip column end, and then we put the emitter tip, and then the liquid mobile phase with the metabolites is sprayed like this environment. And then the solvent and water evaporates, and then only ionized particle can enter the mass spectrometer. In that case, there are 2 modes I mentioned earlier. So usually we use acidic eluent. So that means there are additional protons are existing there. So during this evaporation, for example, this molecule can get additional proton, and then overall this converts to positive ion. So we will use the positive mode to detect this molecule. But that's the case, we will see increase of the 1 Dalton from the original molecular weight. And the negative mode is for octanoic acid, and the molecular weight 144. And if this is ionized and then is a 8 form, and we will measure this molecule in 143 MOG values. So based on the voltage what we put here, we can measure positively charged one or negative charged one. One inconvenient thing for ESI, the electrospray ionization, is we need to run them twice to cover all the metabolites. So positive mode and negative mode, because based on their chemical property, some metabolites wants to be positive mode, and then the others wants to be a negative. So that's a kind of a handicap for the ESI, but it's very sensitive because all the water solvents will be evaporated. And mass analyzer, you may heard orbit trap, time of flight, or ion trap, orbit trap, or other traps. So I cannot cover everything detail, but I just chose a couple of things here. So quadrupole is like we have these 4 different pores here, the metal pore, and 2 of them are charged is alternating current, and other 2 are charged with a direct current. And so inside of this pore, there are electric fields oscillating. So if the metabolites MOG values are well harmonized with this electric field, they can pass this quadrupole. Like this, they oscillate inside and they can detect. However, metabolites, they are not fit within this oscillating direct current and alternating current. They will not reach to the detector, and then they will be just disappear through the vacuum system. And then if we refocus on delta metabolites, MOG values, it only can pass this quadrupole. And also yesterday, Yuchen mentioned about the triple quadrupole. That means we have this one quadrupole and the collision cell, and then another quadrupole at the end. So we can just specifically focus on certain ion. At the beginning, we just screen certain metabolites, and then Q2, there are collision happening, and then later we can specifically focus what fragment ion we can measure. And second thing is time of flight. So it measures the real time, how long the metabolites flew from this point to the detector. So there is this measurement is a little bit based on the pulse. So when we put metabolites and accelerate to at this point, and then we don't give any electric field, and then it has a power and energy still keep flying. So based on this, the lighter molecule reach to detector early, and the heavy molecule will reach later. So we can measure the time difference, and then we can show them as MOG values. And ion traps is as said, is trapping ions. So this is ion trap. It's very traditional ion trap what I used when I was a graduate student. And if ionized molecule enters the ion trap, and this will undergo the oscillation and trapping inside, and then at some specific point, it can pass the exit cap and go to the detector. So there are a lot of variations, quadruple ion trap or linear ion trap or orbit trap. And yesterday, yeah, Christine and Yuchen mentioned about the Lumos that has a hybrid or tribrid. Is there are quadruple and orbit trap and linear ion trap. So many ion traps are located inside one machine. That is a tribrid, the Lumos machine. And then the high resolution, the real high resolution, the Fourier transform ion cyclotron resonance is, we have the 21 Tesla FTIR, and yeah, this will take a long time to explain everything, but simply it is under magnetic field, strong magnetic field, and also we put alternate current and direct current, and then they are usually at normal stage, the metabolites oscillate inside of this chamber, but when a certain environment, certain condition, it hits the detector plate in the outer surface, and then it gets the signals, and then we transform the signal, and then we can measure their M of G values. This is a really high resolution, so we can get accurate M of G values at 5 or 6 decimal point. So if that value is really accurate, we don't need a really long time separation, because a lot of unique molecule has unique M of G values. So far I just covered the chromatography and mass spectrometry here. And Natalie showed a simple thing. So I will not cover much detail about this process, but we need extraction of metabolites from synthetic cell culture. For synthetic biology, the bacteria or fungi, we can extract metabolites and we can measure them based on what we want to analyze. And especially for the environment samples, we need a more kind of extraction step, need to focus to get all the metabolites out from the matrix or soil or microbial mat or such thing. And then I want to talk about the intracellular versus the extracellular metabolites, especially for the synthetic biology research. A lot of metabolites exist in the outside, but we don't know what's happening inside if we just measure the extracellular metabolites. That's why we want to measure both intracellular and extracellular metabolites. So this is the TEM images, what recently we got from EMSL about the Aspergillus niger. And this is fungi, filamentous fungi, and we can see their structure. This is a 20 micrometer, and this 1 micrometer scale bar. And we see something happening inside. This is might be some small sub-organelles inside of the fungi, and they secrete something to the outside. Also, they consume glucose or other nutrients to grow inside. And then a lot of metabolism happening inside. And if, for example, 3-HP, we hope this strain continuously secrete 3-HP molecule what they made. If not, if they accumulate a lot of inside their body, I think that will be toxic to certain, if the concentration reached a certain level. And also the synthetic biology field, we start with agar plate or flask and small bioreactor and the bigger bioreactor. We need to monitor the unwanted metabolic carbon sink or byproducts. Because if we know what's happening, and then if we can address those problems, and we can reach a better production efficiency and to maintain the stability of the bioreactors. That's important for the scale-up process. And one of the metabolomics workflow for GCMS, we need a chemical derivatization, because a lot of metabolites are hydrophilic, and that means they have a higher boiling points. So if we modify their hydrophilic moiety like amine, hydroxyl, or carboxyl groups to nonpolar structure, those metabolites will be vaporized at lower temperature. So for example, this is alpha-ketoglutarate or 2-oxoglutaric acid is one of the TCA intermediates. And our first methoxyamination step, this methoxyamine will substitute this ketone. So it will convert like this structure. This is important because these ketones are reactive, and we increase the temperature, it can react with other moiety in other metabolites. Also, it can be degraded, self-degraded. to smaller molecule. So this step is essential for ketone metabolites. And then after this first step, we use the MSTFA or BSTFA. So this the trimethyl cellular group will protect this carboxyl or amine or this the hydrophilic group. So the final form, what we measure at GCMS is this one. This is the alpha-ketoglutarate in derivatized form. So overall, this will increase the volatility and reduce the hydrophilicity. And also, what we can do by using GCMS is a volatile metabolites. So far, the global metabolomics by GCMS, we need to dry them after extraction. So during that process, a lot of volatile metabolites will be gone. So we are not able to measure them. However, if we use a polar column and then if we inject the sample directly without any drying process, we can definitely measure such as very small metabolites or molecules. So bottom one is one of the examples. We collected microbial mass samples from Yellowstone National Park hot spring. It's very hot. It's about 60, 65 or 70. There are a lot of bacteria, cyanobacteria live there. And we freshly collect and then we kept them in the liquid nitrogen on site. And then we brought them to lab and then we just extracted with a methanol chloroform. And then without drying, we just added isotope labeled internal standards, the 13C acetate, and then we measure their abundance changes day and night. So for example, the acetate and propionate is very 2 carbon or 3 carbon metabolites. And then their abundances are going down during the daytime, and then it goes up in the nighttime. Also, we can inject a lot of microbiome research. We can get fecal or intestinal material, and then we mix it with methanol, and then we can just inject to the machine, and we can measure a lot of short chain fatty acids or other smaller volatile metabolites as is. So Natalie covered these things, targeted or global discovery metabolomics. So targeted is a really hypothesis-driven approach. So we know what we want to measure or their relative the related metabolites. However, in the global approach, we don't know what's going to happen, or we want to see some big changes after smaller alteration of genes or pathway. And so synthetic biology field, we use both the global and targeted metabolite analysis. Is it really focused or kind of a approach? And databases are important and will be covered in the afternoon. And global approach is really based on the database. The targeted is we already know what we want to measure, and in that case, we have the standards, so we can really know what we will measure. However, the global metabolomics is really based on the metabolomics database, and the curation is important. And in-house for LCMS or GCMS, especially for GCMS, is a more reproducible lab to lab or machine to machine. However, LCMS, there are a lot of variations in the column length, flow rate, solvent composition, pH. So LCMS side is a little bit hard to generalize all the protocol. That's why in-house database or library is needed to generate accurate metabolite IDs. So that is a key component. There are some softwares or database or online database, or there are a lot of options. And currently PNNL we have been developing for GCMS or LCMS, and we still keep adding new metabolite information to our library, and then so we will get expanded coverage getting year by year. And this is about the high resolution versus low resolution. If we can measure this M of G values as a high resolution, we can minimize the possibility, because if we just use a single quadrupole mass spectrometer, there are a lot of other compounds are possible. So for example, if we just use a quadrupole mass spectrometer, and then at given M of G values, there are 4,000 of possible combinations of chemicals are possible. However, if we use a time of flight mass, we can dramatically decrease the possible chemical compositions. So even if we use a FTIR or high resolution orbit-trap, and then we can get more accurate M of G values, and then it can increase the correct identification possibility. And also for the GCMS side, we have a orbit-trap GCMS, and all the fragments are also acquired in higher resolution, so we can really increase the accuracy of identification if the metabolites are fragmented. And another thing, I want to show the mass resolutions, and this is one of our recent project collaborating with the University of Washington, and they are seeking for nitrogen isotope incorporation in the metabolites. This is an example of putrescine, and one of these nitrogen got incorporation of the 15N, and our mass spectrometer can distinguish this 15N versus 13C, because if we measure this mass spectrum in low resolution, we only see 175. However, in an orbit-trap high resolution mass spectrometer, we can distinguish these two peaks. And theoretically, the difference between the 14N and 15N is 0.99743. And the theoretical delta M between 12 carbon and 13C is 1.00335. And what we measure here, the peak delta M is 0.9971. So this 175 is definitely addition of a 15N. And the small the peak isotope, the natural one, and the delta M is 1.0026. So high resolution mass spectrometer can, amazingly accurate, and then we can measure them really accurate manner. So data processing side, we will get this metabolomics data, and then we can do simple t-test or ANOVA for the statistical analysis, and then we can do heat map and principal component or PLSDA analysis, and also we can do pathway enrichment or other biological interpretation. So this will be covered tomorrow and the day and Friday. And really the last slide topic I want to mention today is about quantification. So in general metabolomics field, we can do absolute quantification based on analyzing the authentic standards. NMR can provide better data without any standard analysis, but mass spectrometer has all the mass spectrometer have different response factor and different ionization. So we have to analyze the authentic chemical standards. So in general, there are two different ways for the calculation of the real concentration. So the very simple thing is external calibration methods. So we can just prepare serially dilute the standard sample. So we can inject 1 microgram or 10 microgram, 100 microgram, 200 microgram, and we can get corresponding peak area of the standards run. And then we can make a calibration curve. And then we can learn the target sample, and then we acquire the peak area values. So if the peak area value is about 4,000, we can calculate that, oh yeah, there are 80 microgram was there in the sample. And then internal calibration method is a little bit different with this external calibration. And we need to spike internal standards before extraction or before analysis. So for example, we can spike certain internal standard. This could be a natural one, but should be not exist in the sample, and or safely we can spike 13C or 15 and label the internal standards. So we know how much we add in the beginning, and we can also get internal standards runs as a separate. So we can make a 1 microgram for internal 100, 10 to 100, 100 and 100, 200 and 100. So you can calculate the ratio between the internal standards and native. And then we analyze the sample after spiking internal standards. And then we can calculate the ratio. So if the ratio is like 0.15013, then we can calculate this amount concentration is 80. The difference between the external and internal is if we lose some samples during the sample preparation and processing, this result could be a little bit inaccurate. However, even though we have some sample loss or some issues in the instrument, however, the ratio is constant. So we can get better the quantification data. So, and also if the samples are spent in medium, we can easily calculate their volume. So we begin with 1 ml, then we can calculate their real concentration. However, intracellular metabolites is a little bit challenging to generate the concentration. So I prefer to use the cell dry weight. That is the most preferred method. So we can calculate how much metabolites there based on how much the dry weight of the biomass. That's how we generate the metabolomics data for the, especially for the absolute quantification. So summary. Oh yeah, it's just 10.3. So I cover the fundamentals of chromatography and the fundamental mass spectrometry and the general metabolomics workflow and other additional application and the difference between the global and the targeted and a few subtopics. So that's my end slides. And if you have questions, I haven't seen anything, but yeah, I will address, or later I can reply to all the questions in the Discord or this Zoom window. Since the time is slightly over 10:03, thank you for your attention. Yep.
Yeah, there was a lot of questions there, Young Mo, but I think Will and Natalie has been answering most of them. So I think we're over time. Maybe it's time for the break.
Yep, that's good.
Okay, so we come back at 10:30, right? 25 minutes from now.
Thank you.